this afternoon comes from colleagues at the Safer Medicines Trust. So Kathy Archibald, Robert Coleman and Tamara Drake co-wrote this chapter together. But today Jan Turner will be presenting the work as the new Director of Safer Medicines with a short introduction from Kathy. So here you are, Kathy.
Thank you very much. So Safer Medicines Trust exists to try to change the old paradigm of using animals as proxies for humans to a new by showing that a new paradigm based entirely on human biology is better. So it was a privilege for me to write a chapter for this book with Tamara Drake from the Centre for Responsible Science in the US, who's doing great work to try to change the wording of the regulations to encourage the use of more human-relevant technologies, and Bob Coleman, who co-founded the world's first drug discovery company focused exclusively on human tissues. Bob would have loved to be here today and speak about our chapter, but sadly he had a prior engagement. So I'm delighted to introduce Jan Turner to speak on our behalf, because I'm even more delighted to say that she has taken over from me just recently as the Director of Safer Medicines Trust. It's always been my ambition to have Safer Medicines Trust led by somebody with high-level experience of the paradigm-changing technologies that we champion. And Jan has worked with international teams of scientists and regulators on a major project to move human-relevant cardiac safety testing into regulatory use. So my profound thanks to Jan for being here and speaking today, and a very huge thank you to Kim and Catherine for pulling together such an excellent event and such an important book. The paradigm is changing already, it's inevitable, but it's not happening as fast as all of us would like. So we hope that this book will help to turn the evolution into a revolution. Thank you. Oh, you don't want that.
Okay, thank you, Kathy, and thank you, Kimberly, for allowing me to talk today, and hopefully I can do justice to the chapter from Kathy, Bob and Tamara. So, let me just... before I get into talking about new technologies, etc., I'm just going to do a bit of background, and I know Lisa this morning gave some of this background as well, so apologies if you're hearing this again. But in terms of safety testing of new medicines, it's probably a bit of a challenge to try and change this, because the testing is often required by governments and is part of the regulatory testing panel that needs to take place for a new drug to come to market. But having said that, protecting patients is a vital goal, and these organisations like these organisations like MHRA, the FDA in the US and the Chinese FDA are part of the whole ethos about protecting patients, and they create the environment in which we have to carry out these tests. So as I say, it does make it difficult to actually challenge the use of non-human animals in safety testings of medicine. But having said that, the question is, and I think it's been shown today in a lot of the previous talks, is do they protect patients? Do animal tests protect patients? And also alongside that, the key questions are, how well do they predict the safety of medicines, and are there any methods that are equally or more predictive than what we have? So before I do that, just a bit of history, and as I say, I think, Lisa, you covered some of this this morning. So in terms of animal testing, post-World War II, a lot of new chemicals, drugs, additives, etc. came onto the market, as it were. And we saw the sort of organisations like World Health Organisations and individual governments set up bodies to regulate these products, these chemicals. But it wasn't until about the late 50s when they really became institutionalised in response to those early drug disasters, such as thalidomide. Excuse me. Now, when that happened, we saw a huge explosion in the safety evaluation testing program. So we saw, my mice have moved up a bit, sorry about that. We saw, 2-week acute studies moving out to whole lifespan studies. We had the number of animals being used increased dramatically. And we had different tests for different types of toxicological endpoint as well. So you saw things for like reproduction, for mutagenesis, etc. And there was also, a real drive to look at species differences, because we knew that toxicities, mechanisms of toxicity were different in different animals. So the probably misguided thought was using different species would be able to cover all of those and make those interpretations in human more relevant. So that was probably back in the 50s, 60s, 70s. In around the 80s, and I think the talk this morning about this, we talked about the European directives. So kind of that's changed, all that campaigning, all that public awareness, I think, did lead to some changes in the European directive. And we saw a real shift towards protection of animals used in scientific processes. And also then the 2010 one really went one step further and said that these tests should not use animals where a non-animal method could be used and achieve the same purpose. So just in case people aren't aware, a quick slide on the drug discovery process. So this is typically a process that's used in many pharmaceutical companies. And you start with maybe tens or hundreds of thousands of compounds on the left-hand side there in drug discovery. And that's then whittled down to maybe 1 or 2 FDA-approved medicines on the right-hand side here. And where and this process can take sort of 10 years, maybe even more. And this estimate was $2.6 billion in 2013. So we're talking huge amounts of money, huge costs associated with coming out with one medicine. And for the purpose of today's talk, what we're looking at really, the areas we're looking at are the preclinical space. This is typically where the animal testing takes place. Phase 1, where it's typically the first in man studies, where they're looking at both safety and efficacy of the products. And also then towards the end, so once the product has been approved and it's on the market, the phase 4 stage, which is the monitoring phase, we can gain a lot of information about the product's adverse effects in humans. So we've got a bigger population there, and this is where we tend to see that reporting. And the FDA have their MedWatch program, the MHRA have their yellow card system, where patients and healthcare professionals can enter the information there about those adverse effects. And so from this data here, the adverse effects, and also from the clinical trials which take place here, we've learned a lot about how potentially animal models don't work, essentially. And some prime examples of these from some of those clinical studies are given here, and I imagine you're quite familiar with these. Oops. So TGM-1412 is a superagonist monoclonal antibody which was used in 6 volunteers in the UK at Northwick Park in the UK. And basically what happened there was they were injected with this antibody and they all experienced what's called this cytokine storm. So it's a hypoallergenic reaction, I guess, to that monoclonal antibody. And basically there was a lot of multi-organ failure, and those 6 people are actually being, some of them lost their fingertips, etc. And they're really being set up now for some problems in their future because of what happened with this trial. The second one here was one in France, and this was a drug that was for a number of uses. And this, Jaraju talked about off-target effects this morning in terms of GM, but this drug actually exhibited off-target effects that were only found in humans. So it had gone through the animal testing and it was actually found in human cells in vitro as well. but this caused neurological damage in 4 out of those 5 volunteers, and actually one of those died. And then the third example is fluoridine in the US in 1993. This is hepatitis B treatment in phase 1 to phase 2, so looking at efficacy and safety. And this caused massive liver failure and multiple organ failure in 5 of the 15 volunteers. 2 of the survivors received a liver transplant. If they hadn't, they would have died. So, really this is quite compelling evidence that the animal studies didn't work, because all of these have been tested in animal studies previously. Now, TGN1412, in the post-investigational report, they looked at, these were tested in 2 species of monkey, and there was no safety signals, no worries there that came through there, but they looked at the homology, and back to Jared's point this morning about genes and expression and things like that. The homology between the monkeys and humans, there was no citations saying that they were 100% the same, which I guess we expect. And actually, subsequent to this, there was some analysis, and the sequence homologies seemed to differ by about 4%. But that was obviously enough in this circumstance to create this so-called cytokine storm in humans. So, I think this is an extra evidence that really we can't use monkeys as substitute or proxies for the human body, really. Lessons from this were that the guidelines associated with doing the first-in-man clinical trials were revised quite a lot. In this case, it was more around dosing levels. So the way the 6 individuals were injected with TGN was that they were injected immediately after each other, and by the time the 6th one was injected, the first one had already started exhibiting these adverse effects. And so there was some advice and recommendations around the intervals, leaving enough time between dosing so that you can start to look at any adverse effects coming through. But 2 key pieces are around characterization of the target biology. So as we said, using monkeys instead of humans, is that relevant? Is that really what we want to be doing? And the translational relevance of that model. So these are key things that came out, lessons that came out of these clinical trials. And I think, we've probably seen today that most people think that animal studies do not predict human toxicity of drugs. And I'm just referring to Jared's papers again, really, because this is a good piece of work where a number of drugs, nearly 3,000 drugs were tested or looked at in terms of human and animal data. And there was some correlation between human and animal data, but it was just too variable to be predicted. And the same analysis, which is probably even more important, is that there was no ability to detect or predict the absence of toxicity, which is what we're talking about when we're talking of safety of our patients. So I think you can say from that animal studies do not predict the absence of toxicity, and so we can't really predict the safety in humans using these models. I'll just touch on what the false negatives, false positives are. So a lot of the trials that fail will fail because of safety. About 50% of drugs fail due to safety, but also efficacy. And within each group you can have false positives and false negatives. Just to clarify what we mean here, false negatives in safety testing is what we're all familiar with, where animal tests miss any side effects that will then cause a harm in humans, whereas false positives are the examples we talked about this morning as well, where there's a drug that's good, that could be used, but is apparently toxic in animals, but is fine in humans. So they're 2 examples of where safety, false negatives and false positives will result in probably erroneous results for safety. And one important one, I think, is chocolate, because it's actually toxic in drugs. So I think that's probably one of the most important ones. Then in terms of efficacy, so in terms of the therapeutic value of those products, false negatives and false positives. False negatives, and I think, again, Lisa, you referred to penicillin this morning. The clearance of penicillin in rabbits was rapid, and Fleming thought that they wouldn't, it wouldn't be able to be used in a systemic way. And another example is a statin compactin, which didn't actually lower cholesterol in rats, but did in chickens and mammalian cells. So there's lots of examples of these where we have false negatives. And then false positives, which is probably the bulk of the things we're talking about here in terms of efficacy, is these treatments that, for quite major diseases, quite profound diseases, cancer, stroke, sepsis, etc., where there's a huge number of products that have gone through the drug discovery pipeline and failed in clinical trials. So you can see those numbers there. So that's just a summary of the sort of positives and negatives of testing. And I guess, just to kind of bring it all together, where we are right now is that animal procedures are still used routinely to study disease and safety, etc. And they're still considered the gold standard. But actually many of those animal procedures have limited human relevance. There are a lot of human relevant non-animal methods around, and I'll touch on those now. And it's, it is widely accepted now, probably amongst us and some scientists, that we should only undertake experiments where the 3 R's principles is adhered to, or at least according to those principles. So I think the questions still remain: how do we identify and stop using ineffective animal procedures? And I'm referring here again to Jared's paper that came out last week, where he contests that, he says that there was a couple of papers reviewed that suggested that the animal models were valid, could be used, but I think the evidence really from those papers was that they offer no benefit in terms of predicting the absence of toxicity. And then I'll touch on in a little while is about the validation and how do we get people to accept these and these new methods, and how do they validate them. So we've heard about some of these products this morning. They can vary in their complexity, and with the complexity comes cost, of course, but you get better understanding and representation of a disease or a condition using a more complex model. But having said that, if you've got a more complex model, your volumes will be less, you'll have lower throughput, and the turnaround time will be slower. But there are a number of things on the market at the moment and available to academics. So simple cultured liver cell lines in this case, slightly more complex ones where there's groups of cells which have extended metabolic activity over a number of days. And then the real complex ones where we're talking about bioreactors or organ-on-a-chip type approaches. And stem cells were mentioned this morning as well. And this is a way we've been able to achieve lots of different individual phenotypes of cells. So you can generate cardiac cells, you can generate liver cells, and really use those to exploit the underlying principles of toxicity in those cells. And spheroids are really examples of these cells with maybe supporting cells which give a more reflective impression of what goes on in the human body. So these are the types of models. So organ on a chip, we mentioned this morning. Oh, I keep pressing the wrong one, I'm sorry. Organ on a chip, which we mentioned this morning. So you can have a number of different tissues or cell types here. So you can have cardiac, liver, connected through with some microfluidics. Malcolm, I think, will probably talk about the quasi-vivo system here, which is a similar concept where you have different cell types linked together. Computer modelling was mentioned, and that's been quite successfully used to look at the exposure of drugs as well as coming up with new models. But what's the actual exposure of a drug in a human? Microdosing, we've talked about a bit as well, where we can add a very tiny amount of drug to a human and study the metabolism by imaging, and this has sometimes been termed as phase 0, so before phase 1 and after pre-clin. And then various other cell models here that have been used to, in this case, hepatopac has been used to predict the liver damage that was seen with phenyluridine. And the TOX21 program, which has looked at over 10,000 chemicals now in vitro cell-based assays to predict end points in humans. And I think the take-home message is that not one test will do everything, and we need to have a panel of those tests really to look at all aspects of toxicity and injury. This is just one example where that's happened. This is AstraZeneca data where they use a number of different assays to look at liver toxicity. But I think one of the great examples that I was fortunate enough to work on with the FDA and HESI in the US was the SIPA concept, which is all around looking at ECGs, so looking at arrhythmias, so irregular heartbeats. And this is a typical ECG trace, and this is what happens when somebody has arrhythmia. So essentially what's happening here is this is your typical ECG trace, and there's this interval called the QT interval. Now, what was thought before was that there was these are ion channels within the heart, and there was one ion channel called HERG that was responsible for creating this T peak here. And what we saw when many drugs were put through safety systems, they tested for that HERG response, blockage of that HERG response, in the belief that that was what made those compounds then pro-arrhythmic. But what this program of work has done is actually look at all the other ion channels as well, because it's not working in isolation, and some of the other ion channels can potentially compensate for that HERG effect and actually knock out the response. So what they were seeing were there were a lot of drugs that were thrown out, baby with the bathwater type thing, because they apparently had a HERG effect. But if they're looked at in the whole system, looking at all the other ion channels as well, you get a better picture of what's really going on in terms of the electrophysiology of the cell. So this was a big exercise. There were 2 stem cell-derived cell lines. There were 10 laboratories involved globally. It took 28 blinded drugs that had low, high and medium cardiotoxicity or proarrhythmia. And basically what this did was come up with a figure of around 87% prediction for this proarrhythmia, which was much, much better than what was done previously. This slide is a bit busy, but I'll try and explain what's happening here. So here there were 2 systems used to measure this. This is voltage-sensitive dyes and microelectrode arrays. This is your control traces in each of those. And then there were 4 types of arrhythmia that were recorded. The type A is the sort of mild type, and then B, C, and D were grouped together into other. So these were the responses that the scientists were looking at. And basically they looked at 7 different predictors, 7 different biomarkers, as it were, or endpoints. And out of the 7, 3 of them were significant. And this is the data from those 3. So where you've got a low prediction of toxicity, you actually see where there's no arrhythmia, you see most of the low ones here. So these are all the 28 drugs. And then when you have the high ones, you see more of them down here, where you have those arrhythmia conditions. So that's a good predictor 1. Predictor 4, they looked at the time, the repolarization time, so the time between the QT wave, and that's often prolonged, as I mentioned. And here again, you see the ones that are high, you can see how this has increased. The response is up to maybe 800 milliseconds there, whereas the low ones, it's in effect sometimes 0 and sometimes even below that. So straight away, and that was at all concentrations of drug. Predictor 7 was at the clinical concentration. So this is probably the most relevant one. This is showing that at the concentration of drug that would be typically used in the human, these high toxicity ones were really being identified here as increasing that time for repolarization. So this has been published last year, and the process itself, where they've got to, is that it's out to basically questions so that they can think about changing the regulations associated with this. But what this has done is really allowed us to see that, you know, there are human in vitro assays out there. They're used in silico modelling as well to recreate those responses in the cardiomyocytes. And because it's, you know, we've understood here the actual components involved in that arrhythmia, they're able to look at all the different ion channels, put it all together, and come up with, you know, some early predictors of what's going on there. And what this has really done is, you know, meant that we've got less false positives coming through. And so you're able then to take drugs that may have been thrown out because they had hERG toxicity, but maybe actually quite efficacious, and we've been able to rescue those. Okay. So in terms of the models themselves, I think there's a I think there's a number of things we need to look at, and, you know, I think this has been covered to a certain extent this morning. They must be mechanistically relevant, they've got to be robust, you've got to consider time, cost, etc., which endpoints to use. But probably the key point here is how do we validate them, how do we evaluate them, and how do we validate them. And, you know, I think there's still quite a few things we need to look at. Oh, I'm sorry. There's still quite a few things we need to look at. You know, there's one of the biggest things is the technologies themselves are advancing really quickly, and really we're not keeping up regulatory-wise. And I think a lot of the current validations are focused on precision, but we really need to talk about species relevance, which I think is the theme of today, really. So the new approach that we suggest we need to do here is to base this on human biology, and we've seen people like the FDA, you know, embracing this type of new approach. Rather than having a fully validated system that may take 10 years to do, look at qualifications, so supporting data that's associated with that. Use things like reference chemicals like they do in the chemical industry to do read across, so similar chemistries can be used to predict the toxicity, etc. And I think, you know, the idea of a pragmatic, iterative approach is key here. So Rebecca mentioned this morning about the EBTC, using selecting drugs whose toxicities were missed and having a test panel, and that's really what we're suggesting here, and base this on the predictive performance on that. So I know Rebecca talked about this morning, so I'll skip this slide. But this is essentially what we're talking about here with the EBTC, is having these three evidence streams, one that looks at the systematic literature review of animal and human studies, taking the ToxCast assays and looking at those and comparing that to adverse events. And what this is able to do is then let us see how predictive those assays are. This is quite a busy slide, but I think it summarises the sort of obstacles we're dealing with. A lot of the regulatory agencies are pretty inflexible in terms of animal tests. And one thing I did want to highlight was Vanda Pharmaceuticals and the FDA. So they put out an open letter about a month ago where they have been asked by the FDA to carry out a 9-month dog study on their new drug that's coming through. They've got 15 clinical trial data assays. They've got 3-month dog and rat studies, 6-month dog studies, I think, and they're being asked to do a 9-month dog study. They don't believe they should, and so they've basically objected to this because the FDA have put a stop on then progress in their clinical trials. So this is just, this is quite frustrating to have something like this. I've run out of time. I think we've kind of covered all these. Adverse outcome pathways were mentioned, and I think that forms a framework for these types of assays and what we can gain from them, how to use them. There's a lot of key points here we need to talk about, but I think, for us, probably a lot of the take-home is around regulatory and having that change and the validation approach. Sorry. I'm just trying to get forward to adverse outcomes. So these are the things we think need to happen in terms of regulatory change, which I think we all know about. I think, the language needs to change, the education of the regulatory people needs to happen, and the timely updates need to happen as well. So in summary, I know I've gone over, there are human relevant methods there that are needed to detect human toxicities that cannot be predicted from animal studies. Many initiatives and roadmaps and tools are emerging, such as the ToxCast and Tox22 type approaches. In vitro methods can be used to predict with good accuracy, but we understand that this is a huge challenge, and I think lack of confidence in the new methods is perhaps preventing this paradigm change, but we're suggesting a new pragmatic approach can demonstrate that they're more fit for purpose than existing models. Funding is a key piece to this as well. We need to make sure that the funding bodies are allowing for this data to be published to support new non-animal models. And I think I raised the question this morning, is it deadlines that we need maybe to get us over the hurdle of this? I don't know, but maybe that would help in terms of driving the changes that we want to see, such as we're seeing with REACH and the Netherlands with their proposal to stop animal testing by 2025. So that's me. Sorry I went over there. Thank you very much.